Hello, people. Welcome to Cell Biology. What do we got? 24, a little less than half. Let's wait a couple of minutes. Kozlowski showed up. Kaczynski's here. Got my Polacks. Actually, Kaczynski, is that Lithuanian? It ends in a Y. Sometimes that's Lithuanian. You're not answering. Abigail, I'm probing your ancestry.
It's Polish.
It's Polish, okay.
Yeah.
I know a woman when I was in college, her last name ended in a Y, Shavinsky. And she said when it ends in a Y, that's Lithuanian, but she's wrong. Okay, as long as we've got time, why don't you bring your faces up here? Let's say hello to everybody. Let's everyone say hello to everyone. See Bailey Lyons is here. Yay, Bailey. Always glad to see Bailey. Diane Johnson is a bit of a, what do you call it? Lurker. She's a lurker. You never see her face. She's there. She's there. You know she's there. Been doing really well in the class. I'm really proud of her. Sophia Martin. I don't think I know Sophia Martin. Bring your face up so I can make fun of it. I'm just kidding. I'm just kidding. Haley Smith, you were in my class last semester and survived. Hello. Everybody survives. Where do we at? We're up to 29. And they're still coming in. By Brian Pinello. One of my favorite Pinellos. I've had a lot of Pinellos. No, I haven't. You're my only Pinello. But I am on an Ethernet cable because of Brian Pinello, and it should not cut out. Where are we? 33. All right, we're going to go ahead and go. I'm still having computer problems. It's the weirdest thing. It's like my computer won't charge unless it's turned off. So if I just plug it in while it's on, it doesn't work. And my battery's getting old, so it doesn't have much time on it. So it may die in the middle of this lecture. If it does, don't go anywhere. I've got my wife's computer over here, and I'll fire that up and finish the lecture from that computer. So don't leave me. Don't leave me. Okay. Today, we're continuing on with our endomembrane system. And so we started talking about the rough ER. We'll finish our rough ER discussion today. And then we'll go on to smooth ER and then Golgi. And then in the next lecture, which will be Thursday, we'll talk about protein trafficking. How does the Golgi decide where things are going to go, and how does it direct things to go where it needs to go? And also endocytosis, exocytosis, penocytosis, phagocytosis, every cytosis I can think of. And then we'll have an exam next Tuesday. So I hope you're ready for that. That is on the schedule. Okay, people, let's start with, what are we starting with? Post-translational modifications in the rough ER. So the rough ER does more than just synthesize or help to synthesize your protein and membrane. Actually, it's not doing the synthesis, is it? It's just kind of inserting in the membrane or putting it on the other side of a membrane. But it's, in terms of synthesis, it's not doing any of that itself. That's all the ribosome. But once the protein is made and it's stuck into the rough ER, there are a lot of modifications the ER does before it passes it off to the Golgi. One of the things it does is a lipid attachment. So this is one of our old friends, the G protein. We saw this in chapter 14, RAC-ROS CDC42. So the G protein is always associated with the membrane. And the reason for that is it's got a lipid that is attached to it. So that lipid being nonpolar needs to be in a membrane. So it's so that keeps the G protein next to the membrane, which is where all the enzymes are that interact with the G protein. So it kind of turns the activation of G proteins from a three-dimensional problem into a two-dimensional problem, right? So instead of G protein floating all over in the cell, wondering where the hell it's supposed to go, it's only floating across the plasma membrane, wondering where it's supposed to go. No, nothing wanders around in the cell. Everything knows where it needs to go. But anyway, lipid attachment, that's another thing that the rough-ER does. Okay. Post-translational modification, adding core glycosylations. So we usually associate the Golgi with glycosylation, and it does do a lot of glycosylation too. But the glycosylations start in the rough-ER. So the carbohydrates that are added for the glycosylations are mannose, glucose, and N-acetylglucosamine. And I don't expect you to memorize these structures. The only reason I put this up here is because I think it's neat to look at. I mean, you look NAG, N-acetylglucosamine, sounds kind of complicated. All it is glucose, except it's got an amine group there instead of that alcohol group. And then it's acetylized, N-acetylglucosamine. So anyway, those are the three sugars that get added. So why is it adding these sugars to it? And a big chain of them. We'll see. Let's go to the next one. Ooh, there it is. Big, huge chain of these sugars. And it's always in the rough-ER. It's always going to be added to an asparagine residue. Asparagine has got an N in its amino acid side chain, and so these sugars are added onto that N. So it's called N-linked glycosylation. Keep that in mind, because in the Golgi it will be different. It's added to different residues. So here in the rough-ER, N-linked glycosylation because it's added to asparagine. Why is it being added? The thing about these glycosylations here in the rough-ER is that it's like a big handle that's stuck onto the thing. So when the protein is being made, it comes into the membrane and gets this glycosylation put onto it. And this glycosylation will bind to other proteins that are specific for it, receptors for it. And that will hold it there in the rough-ER until it's completed and ready to move on. So this glycosylation, a lot of it is about controlling the movement of the protein. So, you know, we're tempted to think the rough-ER, the protein gets made and then finds its way into a vesicle and finds its way to the Golgi. But remember, everything in the cell is controlled, and that makes it a lot more efficient. So this is one of the mechanisms of control. The glycosylations are added to an asparagine, and then you've got glycosylation receptors in the rough-ER that grab it, hold on to it, keep it in place until it's complete, and then they let go, and it gets passed on to the Golgi afterward. In terms of this particular figure from your book, don't bother memorizing this. I think it's really cool. I mean, I was a biochemist as an undergrad, and I think it's really neat, but you know, don't bother memorizing. I'm not going to ask you any specific questions about this. Just know that it's there for control of the movement of the thing. Also, some of this, I'm not 100% sure about this. I've said this for about 27 years now, and it just struck me today. I don't know if it's true. It may be used for immunological recognition. You know, glycosylations in cells at the cell surface are used for the immune system to look at and make sure they're correct, to tell the difference between you and any kind of invading cell. That's true of glycosylations. I'm not sure if these core glycosylations are involved with that or if they're removed in the Golgi. That's definitely true of the Golgi glycosylations, but these I'm not really sure. But it may be part of the process as well. If anybody can find out whether that's true or not, I will give you 3 bonus points. First person with the answer. Okay. That's my lazy way of getting shit done around here. It's like I could search it on Google too or Wikipedia, but 3 bonus points. No skin off my butt. Okay. What else is it used for? proper folding of the protein. So again, we tend to think proteins will just fold up spontaneously, and most of them will, a lot of them will, okay? You can take a lot of proteins and heat them up and they denature, cool them down, and they renature perfectly, right? Not all proteins are like that. If you think about albumin from an egg, you heat it up and it denatures, and then it sticks to itself, and then you got a white egg now instead of a clear egg, right? And it never goes back. So some proteins need help to fold properly. And that's what's going on here in your in the lumen of the Rough-ER with these chaperone proteins. So you can imagine this protein's being made, and as it comes out of the ribosome, a chaperone protein grabs it. And see, as it's coming out, it's got chaperone proteins lined up all along it. And then another series of enzymes will grab onto those chaperone proteins and then fold the thing right, release the chaperone proteins to be used again. And now you've got a functional protein, okay? So that's another thing that goes on here in the Rough-ER is protein processing, making sure that they're folded properly. Next. This is something you would know about if you took biochemistry. Hopefully all of you have, or you will eventually, and that's disulfide bond formation. So cysteines, as you all know, have got an SH group as their side chain, a carbon SH2 group, SH group. So a lot of times in proteins, two different domains are kind of need to be close to each other, and this can be stabilized by putting a cysteine between those two domains and then reducing them to, is that reduction or oxidation? Oxidizing them. So you're oxidizing them, taking out 2 electrons and a hydrogen, and then binding them to each other. Now you've got a covalent bond. So this will keep this protein stable at higher temperatures. So this particular protein is, I believe, hang on a second, RNase. RNase is a kind of a small protein. It's got 1, 2, 3, 4 different disulfide bonds, which means this is very stable. It's really hard to kill this enzyme because those bonds keep it in a good form. So RNase breaks down RNA. And nowadays, it's not a big deal. When I was in grad school back in the late 80s, you could not isolate messenger RNA without going to extreme lengths to get rid of RNase, because RNase is everywhere. It's like it's on your fingers, it's everywhere. Any glassware you have has got RNase on it, so it would destroy your messenger RNA. So you had to go to extreme lengths to get rid of it. Nowadays, they've got inhibitors for it, so it's not that big a deal. But point being, with these disulfide bonds here, you would have to take all your glassware, put tinfoil over the top of it, and bake it at 500 degrees for 3 hours just to kill off that RNase. And then you had what would be considered clean glassware. So stabilizes proteins. Okay, so backing up one to the proper folding, what happens if it's folded? Well, that would just, if it got passed off into the cell, it would just fill up the cell with misfolded proteins, and the cell would be less functional because of that. So it's got this mechanism called ER-associated degradation, ERAD, which is going to have a number of different enzymes. One will recognize the misfolded protein. Another one will add a small protein called ubiquitin to it. And ubiquitin, then, you can see these little circles here are chains of ubiquitin protein added to it. And those chains of ubiquitin will then direct this protein to go to a proteasome, and the proteasome will destroy it. So that whole process of ubiquitin and proteasome destroying proteins, we'll talk about that much later, I think in chapter 23 or 24. But for right now, understand that ERAD involves recognizing misfolded proteins and then adding ubiquitin and having them destroyed. Okay, that's all you need to know right now. But later, when we learn more about ubiquitin, it's basically a really great mechanism in the cell that can pick out specific proteins and have them degraded, even if they're functional or whatever. You can, all right, get rid of this protein, boom, and gets rid of it. So we'll talk about that later. All right. Another thing that's important here in your rough ER is the assembly of multi-subunit proteins. So again, back to your biochemistry. Primary structure is the amino acid sequence. Secondary structure is the curling or beta bending and beta barreling stuff of local structure in a protein. Tertiary protein is how do all those local structures come into a three-dimensional structure. Quaternary is then how do you have separate different proteins coming together to make one big functional complex. So that's quaternary structure. And that's what this 4-0 means if you haven't had biochemistry yet, quaternary. So here's an example. We've talked about this guy already. This is the voltage-dependent calcium channel. So this is the main subunit of the voltage-dependent calcium channel, the alpha-1. But the alpha-1 by itself, it's not fully functional. It doesn't really work right unless you also have a beta subunit, a gamma subunit, and a delta subunit. This alpha-2, I don't think that belongs here, does it? I don't remember. Oh, yeah, it does. Yeah, so there's an alpha-2 subunit too. So 1, 2, 3, 4, 5 different subunits. So the thing isn't fully functional until they're all together. So when the alpha-1 subunit gets made in the rough ER, it is grabbed onto and it's held there until you get the other subunits coming into it. And then once all the other subunits are there, then it can be passed out of the rough ER over to the Golgi and off to the plasma membrane. So that's another project here. Another role here of the rough ER is going to hold these things until the quaternary structure is done and then pass it on. Okay, that's it for our rough ER and post-translational modifications in the rough ER. And after we finished our rough, we're going to smooth. I'm going to smooth, people. Smooth ER. Remember from our last lecture, smooth ER is more like tubes than flattened sacs. And again, you got a lot of surface area when it's tubes like that. So one of the main things that's happening at the surface is the production of more lipids to make more membranes. So whenever the cell has to grow, whenever it has to divide, it's going to need more membrane. So that is done by the smooth ER. And we'll talk in a minute about different functions of the smooth ER that are different in different cells. Right, it's a pretty multifunctional kind of tool for the cell, but that's kind of adaptations to different roles. This is done in every cell. Every cell that's got a smooth ER, the smooth ER does this. So it's making phospholipids and other lipids to make membranes. So your 4 different phospholipids, which you probably should know, but if you don't, I can give you a little intro here, are phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, and phosphatidylinositol. We've talked about PI already in terms of the regulation of microfilaments, the actin microfilaments. If you remember, when you phosphorylate PI, it becomes PIP and then PIP2, and that causes CAP-Z to come off the microfilament and allows it to be polymerized. This is like review for your final, people. It's all here. So anyway, PI we'll see a lot too in chapter 23 when we talk about it being used in signaling. But anyway, these are your 4 phospholipids, and the only difference between them is this group that's added here. So your phospholipid then is a glycerol backbone. This is glycerol with 2 fatty acids on it, okay? And I don't know the names of the fatty acids, so don't ask me. And then it's got this phosphate group to some kind of polar group. So this becomes the polar head group that's always represented by a big circle here. And then the rest is the fatty acid chain that comes down into the middle of the membrane. This is choline. We'll see it when we talk about acetylcholine, if we do. But also this choline can be one of these polar head groups. You can see it's got quaternary nitrogen here, so that's always got a positive charge. It's always very polar. Phosphatidylcholine is the main phospholipid in membranes. And that's just because it's at higher concentration than the other ones. And this production of phospholipids only happens at the cytoplasmic side. Am I right? Or did I make that up? Let me make sure. I think it's cytoplasmic. Cytosolic side. Why do I doubt myself? I'm always right. Okay. Always happens at the cytoplasmic side. And you can imagine if you did that, your membrane would start to curve because you've got more lipids on the outside than the inside. Eventually, you got to do something about that. So the way you get lipids to the other side is to flip it with an enzyme like this, flipase. Okay, so flipase will take something from the cytoplasmic side, flip it over to the luminal side, or backwards. That's flipase. You also have flopase and scramblase, which again shows you that scientists have a sense of humor. Flipase, flopase, and scramblase. So this is how you get, since this is always on one side and this makes it equal. So your membrane is more symmetrical. Now this is how you get membrane to your, well, it starts in the smooth ER. Smooth ER is contiguous with the rough ER, but you could also have vesicles coming off the smooth ER going over to the rough ER. So you've got membranes going back and forth there. You always have membrane going from the rough ER to the Golgi, from the Golgi to the plasma membrane, and from the Golgi to the endosomes and lysosomes and stuff like that. So all of that is just kind of connected by vesicles moving from one compartment to another. Where that doesn't work is when you've got your internal guests, let's say, your house guests who never left, your mitochondria, what do you call that? Endosymbiosis. So to get more membrane to the mitochondria, so you can make more mitochondria, you have to transfer it from the smooth ER to the mitochondria. And that's done by proteins like this. This is phosphatidylcholine PI. Oh, PI is the inositol. Phosphatidylcholine, phosphatidylinositol transfer protein. So it's taking PC from the rough ER over to the outer membrane of the mitochondria, adding it there, pulling PI out and going away. Okay, so it's transferring phospholipids from one to another. And it can be in one direction as well. Here's your smooth ER here, and you've got this transfer protein that's taking phospholipids from here, dropping them off over there. So if your mitochondria needs to get bigger, this is how it happens. And then once it gets big enough, I believe it divides by binary fission, just like a bacterium would. And then you got more mitochondria. Okay, what else do I want to say here? Transfer. So these are called PEP, phospholipid PEP, that's stupid. Oh, I didn't upload these notes to you yet. Let me upload these notes to you right now so you can look at them if you want to. Hang on one second. That's Curly from the Three Stooges. Sorry. I just got to sometimes. Okay. So I'm just going to leave them at the bottom of the first block of material. I need a soundtrack. My life needs a soundtrack. Anyone can come up with a soundtrack for me, you get 1 bonus point. Whatever. So biology notes. Okay, so those notes are in Moodle now if you want to look at them while I'm talking here. Sorry about that. Sophia Martin. I don't think I've seen your face before. Okay. Okay, back to work. Okay, so moving on. In addition to this general role of making new membranes for the cell. This, depending on the cell type, the smooth ER has got different specialized functions. So one of them we've seen already, calcium metabolism. I talked about this in chapter 8 as one of the transport mechanisms. So this is really, really important for the smooth ER in that I think just about every cell I can think of anyway, the smooth ER represents a store of calcium. So it's really low inside, or really low outside and really high inside. And then that can be used to signal the cell to change physiology. We saw that in the muscle cell. When you trigger calcium leaving a muscle cell, the cell contracts. You'll see this in gland cells. You stimulate them with the hormone, calcium comes out and they secrete some kind of something, whatever the gland is secreting. Platelets, you activate this in platelets, they start sticking to each other. We saw, did we see that? No, we didn't talk about that yet. But anyway, bottom line, this is really important for signal transduction. And just about every cell will do this. I'm not sure if every cell does, but every one I can think of anyway, which is basically muscle cells. That's all I can think of. Okay, another thing that's important, and if you had my pharmacology of recreational drugs, I talk about this a lot in that class. I'm just going to go over it briefly here, but in a lot of cells, but especially liver cells, the smooth ER is the place where you've got a lot of enzymes to break things down. So here is your alimentary canal here and your circulatory system. Everything you eat goes directly to your liver. Your liver over here. Everything that gets absorbed by your intestines goes to your liver first through this hepatic portal circulation. So portal circulation is kind of interesting too. It's basically between two organ systems. Normally you think heart pumps blood out to the organs, goes into a vein, back to the heart, right? Well, this is like heart pumps it out to an organ, it goes to another organ, and then back to the heart. So that's what portal circulation means. Maybe you learned that in A&P if you had A&P. But you see, you pay for one class and you get 3 or 4 here. You guys are lucky. Okay, so anyway, anything that gets absorbed in your GI tract gets over to your liver first. And that gives your liver a chance to detoxify it because, any kind of plant you're eating, less so animals, but with plants, sometimes plants don't want to be eaten. So they've evolved toxins to make you sick. So you eat that plant and you're like, ugh, I don't eat that anymore. Well, your liver can save you if you eat too much of some toxic plant because that chemical will go to your liver will break it down, and then it goes into your bloodstream and your kidneys can get rid of it. So this is one of the enzymes involved in that. It's actually a series of enzymes. There's a whole bunch of these, cytochrome P450s. So these will break down unknown organic chemicals as a mechanism of detoxing. And usually what they're doing is converting a chemical from something that's non-polar into something a lot more polar. And it's a general mechanism, which means sometimes it's going to turn a chemical off, sometimes it's going to turn a chemical on. It's not specific, but in general, it's turning things off and getting rid of them. It's got 2 reasons for making it more polar too. One is your blood-brain barrier. Okay, so here's blood that is feeding your brain. For something to get into your brain, it has to either be non-polar so it can diffuse out through this blood-brain barrier, which is basically lipid wrapped around the capillary, or there has to be transport mechanisms. So glucose gets transported into your brain because your brain needs glucose. But if you ate some toxin that there's no transport system to, your liver turns it non-polar and then it can't get into your brain. What happens then is eventually it makes its way to the kidney, and if it's polar, the kidney can get rid of it. So the kidneys get rid of polar things. Okay, so that's detoxification. What else? I'm glad you asked. You also have carbohydrate metabolism. Okay, I'm not sure how important this is. I'm sure it's important, but I don't know how widespread it is in different cells, but it definitely happens in the liver, where your liver stores a lot of glycogen. So again, hepatic portal circulation, liver, your food goes to your liver. If you've got a lot of glucose in your food, a lot of sugar, it'll change that into glycogen to store it. And then your blood glucose drops, your liver releases glucagon. Glucagon causes glycogen to be broken down. And then in the process of breaking down glycogen, it ends up phosphorylated. Now, remember our glucose transporter from chapter 8, it doesn't transport phosphorylated glucose, right? It only transports regular glucose. So one of the things your smoothie R is going to do is it's got this phosphatase enzyme here that will take that phosphate off and allow glucose to leave the cell. So there's the smoothie R is involved here in carbohydrate metabolism. And then another really important process is steroid biosynthesis. And by steroid, I mean more than just hormones. I mean cholesterol. Cholesterol is an extremely important molecule for us warm-blooded animals. It goes in the membrane and helps to modulate the fluidity of the membrane. If the membrane gets too stiff, your membrane proteins stop working. If it's too fluid, they stop working. So this helps to modulate that fluidity to keep your membrane proteins functional. And it is made in the liver. And then cholesterol then gets transported around the body in low density lipoprotein particles, LDL, and then any cell that needs it will pull those LDL particles in and use the cholesterol. We'll talk about that in the next class with receptor-mediated endocytosis. So anyway, you can imagine your favorite gland. I know this is your favorite gland, the cortex of your adrenal gland, or the adrenal cortex. And this will suck in LDL particles and convert cholesterol into corticosteroids. Corticosteroids are important in electrolyte balance, which is keeping your blood pressure right on a long-term basis. So if you lose sodium, your blood pressure will come down. If you reabsorb sodium, your blood pressure goes up. So your adrenal cortex is really important in blood pressure control, and that's because of your corticosteroids that are made from cholesterol. Also, you've got your other your sex hormones, testosterone, estrogen, progesterone, and I believe there are other things, other corticosteroids that are important, but I'm not an endocrinologist either, so we're going to let that go. One thing, as a pharmacologist, as a cardiovascular pharmacologist, I want to tell you about is this series of drugs, the statins. I don't know if you've heard of these, but just about everyone over 60 is on these. I'm not, because my lipid profile is really good. But if you've got high LDL, that can be really bad cardiovascularly, because it starts an inflammation process in the arteries to your heart, and then the arteries close down, and then you have a heart attack and die. So what these drugs do is block this HMG-CoA reductase, which is the rate-determining step of cholesterol biosynthesis. Let's put bio in there just to be bio. So here's the thing, you go into your doctor, they do a lipid profile, they say, oh my God, your LDL is off the charts. We want to put you, or your LDL is a little high, so we're going to put you on a low cholesterol diet. So what happens to your cholesterol when you go on a low cholesterol diet? Nothing. It'll drop a little bit, but not much, because your liver is controlling your cholesterol, not your diet. So if you totally take all cholesterol out of your diet, your cholesterol level drops, and your liver says, uh-oh, we don't have enough cholesterol, and it makes more. So your cholesterol is controlling that. So if you've got high LDL, the way to get it down is to block this with the statin drugs. So you block this rate limiting step, cholesterol can't make liver can't make cholesterol anymore, and your cholesterol comes down. And by the way, this is another herbal medicine. These were adapted from an herb called red rice yeast. I guess it's not herbal. It's yeast, which is a fungus. But anyway, that's where these drugs came from. But anyway, I digress. Dr. Slish, why does everything have to do with cardiovascular health? Because that's what I got my PhD in. God damn it. Damn you kids. Okay. So that's it for the smooth ER. You've got your lipid biosynthesis and then 1, 2, 3, 4 different specific things based on the cell you're in. Okay, and you can think of the smooth ER as off to the side. It's kind of like a sideshow. Important, of course, but sideshow. The main show here in this chapter is the rough ER and the Golgi working together to make proteins and send them off to their destination. So back to your main theme here, the Golgi network. So the Golgi network is a whole bunch of flattened sacs. Okay, so again, it's kind of like the rough ER, and then it's got a lot of luminal space and a lot of big sacs. And vesicles will come into this Golgi network from the rough ER at this face. And so this face of the Golgi faces the rough ER. This is the cis side. This side is the trans side. It's the side away from the rough ER. And then the middle is medial. So cis, medial, trans. And let's get another picture. Oh, there's the electron micrographic. Okay. So here's your Golgi, a bunch of sacs. Here's your rough ER over here. So vesicles will bud off the rough ER and then move over to the cis side of the Golgi, fuse there, and then any proteins that were in the lumen of the rough ER will now be in the lumen of the Golgi. If there are any proteins in the membrane of the rough ER, they go into this vesicle as well, and then they become part of the membrane of the cis Golgi network. Now, inside this sac, you've got a bunch of enzymes, which are going to do post-translational modifications of those proteins. And you've got different enzymes here versus here. So you've got a series of reactions that happen to these proteins as they pass from one sac to another. So every sac's got a different series of enzymes. And that happens by just vesicles coming off one sac of the Golgi and onto the next, off of that to the next, off of that to the next. So you've got vesicles transporting proteins from one end to the other. Once it gets to the trans Golgi network, the protein is completed. It's got all these post-translational modifications done, and it's been given a tag that tells it where it needs to go. Some things then from the trans Golgi network will head off to the plasma membrane and exocytose. Why is it doing that? Well, if there's something inside the vesicle that needs to be secreted from the cell, that's one mechanism of, one way to do that. Send it over to the plasma membrane, it fuses and releases. So you can think about like breast tissue. When prolactin is released and it goes to the breast tissue, stimulates those secretory cells to start making milk proteins. So milk proteins get made in the rough ER, pass, and they exocytose into the ducts, and then the ducts, lead it on. You could also have vesicles that are made like in a nerve cell that are not going to immediately fuse, but they're going to sit near the membrane and wait for the signal to fuse. And we'll talk about that in the next class as well. So that's another secretory mechanism, was it's not like obligatory release, but controlled release once it gets near the membrane. Another reason for this exocytosis is not to release what's inside the vesicle, but to get membrane proteins that are in that vesicle into the plasma membrane or some other membrane in the cell. It could be even the smooth ER or back to the rough ER or whatever. So that's another part of this exocytosis mechanism, is getting those membrane proteins to where they need to go. Another thing happening over here in your Golgi is the production of these early endosomes. Okay, so these are endosomes. So endosomes then are small, are vesicles that are filled with digestive enzymes, acid hydrolases. So these are not, pancreatic and digestive enzymes. They would be released by exocytosis. These are intracellular digestion enzymes. And they're only active in acidic medium. So if these endosomes open up and release in the cytoplasm, those enzymes don't destroy the cell. Okay, they're inactive until those early endosomes then will fuse with vesicles that are being endocytotes. So a lot of cells will, grab food from the outside world by endocytosis, or bring in important particles by receptor-mediated endocytosis. And those vesicles then will fuse with these early endosomes to become the late endosomes. And the late endosomes will then acidify using that proton pump from chapter 8, and then it's a lysosome. And now the lumen of that lysosome is acidic, and that activates those acid hydrolyses. So proteins that are involved in the lysosome then will be tagged to go into these early endosomes, and then those early endosomes fuse with endocytosed material and eventually become lysosomes. We'll talk about that tagging in the next class as well. How do you make sure that this protein that was made way the heck back here ends up here and only here? We'll talk about that. Okay, so another aspect of this Golgi is some things get passed backwards, even back all the way back to the rough ER. So if we're going forward, we call that anterograde. If we're going backwards, we call that retrograde. Let me do something real quick. Move that link because it's bothering me. Okay, so retrograde transport. Why are things retrogradally transporting? Some things from the lysosomes will be retrograde passed back. I'm not really sure why, but it does happen. More importantly, or at least what I know about, is as you're passing things from the cis to the medial to the trans, sometimes enzymes that really should be back in the cis end up in the medial. So this retrograde transport brings them back and puts them into the former compartment. Or if you remembered when I was talking about the rough ER, the rough ER has got these receptors there, and you glycosylate some proteins and they attach to them. And then that can be used as a mechanism to drag that protein over to the cis Golgi. Once it's there, you got to get that receptor back, right? So you have a receptor that grabs a protein, brings it into a vesicle, fuses with the cis Golgi network, releases that protein, and then has to be transported back to the rough ER because that's where it belongs. That's its function. So we'll talk about trafficking in the next class, but that's a lot of what's going on there. Okay, so Mr. Golgi is involved in finishing off proteins. So they're coming by these transition vesicles to the cis. They get passed from sac to sac through the medial to the trans. From the trans, they go off to wherever they need to go. And some things can come retrograde back, but most things are going anterograde forward. How are these vesicles moving? Glad you asked. It's always tempting in the cell because in biology, because it's just easier logically to think, oh, this just floats here because it needs to go there. Nothing is accidental in a cell. Everything's controlled. So what's going on here in this anterograde transport is these vesicles have a motor map attached to them. If you can remember back to chapter 14, that motor map is then put on a microtubule track and it's brought forward until it fuses with this Golgi. We'll talk about fusion in the next class too. That's not automatic either. There's proteins involved in that. But all these movements of vesicles are due to motor maps attached to the vesicle, dragging it along a microtubule. Okay, now, two different hypotheses about how a Golgi forms. One is called the stationary model, which is kind of what I've been saying up to now, which is you got your cis-medial trans, vesicles move in this way, move through and move back out. Some vesicles move backwards to keep the enzymes in the right compartments. But pretty much your cis is your cis, your trans is your trans, and vesicles are used to move things back and forth through it. The other model is you get these vesicles coming from the rough ER fusing together. And once they fuse together, they form a cis Golgi, okay? And the cis Golgi, the sac itself kind of transitions forward, and enzymes are passed back and forth by these vesicles. So you've got enzymes that should be earlier being passed back this way, retrograde, and the anterograde is pretty much the whole sac changing as it goes on. And then this trans-Golgi network, the sac just dissolves into vesicles and they go off to where they need to go. So that's the cisternal maturation model. And apparently there's evidence for both models. It's probably both are being used to a certain extent in different cells. So that's just two different models of how the Golgi is controlled. Okay, what are the roles? What post-translational modifications are happening inside the Golgi? Again, thank you for asking. There are a lot of different enzymes that are doing a lot of chemical modifications. I didn't write this in the notes. I should. I think I will and then repost. So we always think of the Golgi in terms of glycosylation. I'll talk about that in a minute. But you also have other things going on. You've got phosphorylation, sialization, putting sialic acid on. You've got proteolysis reactions happening. There's a lot of different reactions that happen inside the Golgi. We always focus on the glycosylation because it's the most obvious and easiest thing to do. But keep in mind there are other things that happen in the Golgi as well. But anyway, getting back to glycosylation. So glycosylation here in the Golgi is going to be on different amino acids than in the rough ER. In the rough ER, it was always on asparagine, so it's N-linked to that nitrogen at the end of the amino acid. Here in the Golgi, it's called O-linked because it's going to be added to serines and threonines. And serines and threonines have got an oxygen as their side chain, an alcohol as their side chain. So the sugars are added to that oxygen. So it's called O-linked. What else do I want to say about that? Okay, and it's always going to happen on the luminal face of the, always occurs in the luminal face, this is wrong, of the Golgi. Just correcting my notes, I'll re-upload later. So this is always happening inside the lumen. So the enzymes for modification of these proteins are in the lumen. So the luminal side of the protein is what's being changed. That's important. Remember in the last class, we talked about how whatever's in the lumen will be extracellular if it's put in the plasma membrane. Whatever's cytoplasmic will stay cytoplasmic. So glycophorin, we talked about in the red blood cell, it's one of the proteins that the cytoskeleton is attached to in the red blood cell. This would be in the lumen of the Golgi, and it gets a lot of glycosylations on it. And then when this is put into the plasma membrane, it becomes extracellular, and that's used for immune recognition. So definitely these glycosylations are important for immune regulation in the mammalian cells. And again, we're talking, the sugars we're adding here are mannose, N-acetylglucosamine, and glucose. And these are called terminal glycosylations because these are the end product. Some of the ones from the rough ER will be removed in the Golgi, maybe all of them for some proteins. And some glycosylations will be added in the cis and removed in the medial. There's a lot of changing of the glycosylations as you go through. And again, a lot of that has to do with trafficking. How does the cell make sure that the protein keeps going through and doesn't get stuck in the cis? Well, it puts a glycosylation onto it, has a receptor that grabs it, moves it to the next. The receptor lets go of it there. The receptor moves back and keeps doing that. So there's a lot of trafficking that goes on because of glycosylation. But once it gets to the trans Golgi, it's got its terminal glycosylations and it's ready to be exposed to the extracellular medium. Media, plural, medium, singular. Just teaching a little Latin too. You guys are so lucky. Okay. So here are your O's. So rough ER, this is N-linked because it's added to this N of the asparagine. Here's Golgi, O-linked. Here's serine, and here's threonine. So we're attaching to the OHs here. And here's your N-acetylglucosamine again. And here, again, don't memorize these pathways, but you can see as you look at the rough ER, what are the things that are happening here? You should read through this. Although, again, I'm not going to test you on it, but see how things change as you go from here to here to here to here. Okay. Okay. And this bad boy, I want to save for Thursday. We need something to talk about on Thursday, right? Right, troops? Okay. All right, so I guess that's what I have for today. Anyone have any questions, comments? Any discussion? Brian Pinello, bring your face up, Brian. See?
Damn, I wish I could, Dr. Slish, but my freaking laptop doesn't have one. You know, I had to be economic in these times.
You do. Yeah. Save your money. No one wants to see you anyway. Save your money.
Yeah, pretty ugly, so that's pretty beneficial.
I disagree. You're a good-looking man. You're a good-looking man. I'd date you if I was gay. I would.
That's just kind of my hair. That's just because of my hair. You just like me because of my hair.
Oh, man. I'm going to have to edit out that I would date you thing before I post this because I could probably get arrested for that. All right. Anybody have any questions, comments? So Thursday we'll get together. We'll finish this chapter, and then next Tuesday is the final. If we end early on Thursday, which we probably will, I'll do a review for you people. Bring questions with you. Tell me what you don't understand about the chapter, and we'll talk about all of that on Thursday. And then Friday, I take the day off and you guys work, right? All right. Have a great day, guys. We'll see you Thursday.
You too. See you Thursday.
Okay. Bye.